There is a line in an FDA workshop slide deck that should change how you look at your own drug substance. Structural signatures are molecular fingerprints left by the manufacturing process. Not your specification, not your assay, your process written into the molecule where a reviewer can read it. Hello, I'm Raghuvar Singh Parmar, Director Scientific Affairs at Resolve Mass Laboratories. We work with pharma and biotech teams to build the characterization data that supports active ingredient sameness in complex generic filings. Today, we'll look at what a process signature is, which material carry one, and how FDA awards the deficiency when the data falls short. Before we start, I would request you one thing. If you have found anything valuable from this channel, please like this video and subscribe this channel. This gesture of appreciation would really means a lot to us. Now, let's start with heparin. In 2008, a contaminant called oversulfated chondroitin sulfate reached the supply chain, and compendial tests in use at the time did not separate it from heparin. USP revised the monograph and added proton NMR and capillary electrophoresis, two methods that read the shape of molecule instead of only counting activity. That moment taught the industry something it still builds on. For a complex material, the number on the certificate of analysis is not the whole identity. The chemistry that made the material is part of the identity too. So, what is a process signature? It is a structural feature in the drug substance that exists because of how the material was made, not because anyone designed it. Enoxaparin is the clearest case. It is made by alkaline depolymerization of a heparin benzyl ester, and that chemistry leaves a 1,6-N-hydro ring on the reducing end of about 20% of the chains. The label carries it as the range 15 to 25%. That change the depolymerization route and change number, and that number really moves. The molecule keeps a record of the reaction. So here is the problem that creates. For a complex mixture, no single method sees the whole molecule. Chromatography gives you distribution, NMR gives you linkage and sulfation pattern. Mass spectrometry gives you sequence and fragment maps. Each one is a partial view. FDA's 2022 draft guidance on sameness evaluation says it plainly. Use orthogonal methods to characterize the compounds, and the agency will weigh the totality of evidence. Orthogonal is not a buzzword here. It means methods that fail in different directions, so what one misses, another catches. Where the signatures gets written. So where in the process does the signature really gets written? Three places: the reaction route, starting material, initiator, reagents, then the polymerization or depolymerization step, which sets chain length and cleavage preference, and then purification, which is the one that is easiest to overlook. In FDA's glatiramer acetate work, the ultrafiltration and diafiltration step does not change any peptide molecule, but it changes which molecules are still in the mixture. So the collective fingerprint shifts. A cleanup step moved the signature. If that point is making you look again at your own downstream steps, a quick like in this video helps really us to understand the things. Now, which materials carry the most risk? Not every material carries the same weight of the signature. At the top sit random copolymers and colloids like glutationic acetate and iron carbohydrate complexes, where the whole complex is the active ingredient and there is no sequence to line up. In the middle are low molecular weight heparines and polymeric excipients such as PLGA, where the lactide to glycolide ratio, the end cap, and molecular weight distribution all move with the process. At the base are highly purified synthetic peptides. You can sequence those, but the signature still lives in the peptide related impurity profile. Now, 3 fingerprints and 3 chemistry. So 3 real examples show what reviewers actually read. For enoxaparin, FDA approved a generic through an ANDA on a 5 criterion sameness package, and criterion 2 is the heparin source material and the mode of depolymerization. The process listed as a sameness criterion. For glutationic acetate, the criterion 3 is titled Equivalence of Structural Signatures, covering initiator uptake, propagational shift, and preferred cleavage sites. For iron carbohydrate colloids, it is core size, crystallinity, and how particle size distribution behaves on dilution. At Resolve Mass, we have supported sponsors whose comparative characterization began as a short method list and grew into a full orthogonal package once the reference product data come back. Now, how do you build the package? Map the root first and write down which attribute each step can move. Characterize the reference product across multiple batches, not one. Run tests and reference side by side, same day, same instrument, same reference standard lot. Add orthogonal methods, so no single techniques carries the conclusion alone, and you have multiple data to compare and conclude an orthogonal approach. And then the step that is easiest to leave out, negative controls. FDA's glutationic deficiency list names improper negative control studies, material made off target on the purpose to prove the method can see the difference, because the finding is often not the product is different, it is that the data could not have shown a difference either way. So let's talk about key takeaways. So what should you walk away with this content of the video? A process signature is structure left behind by chemistry. No single method reads it. So orthogonal data and side-by-side design carry the argument. Materials with no sequence carry the most risk. And method that cannot fail a wrong step cannot really pass a right one. Thank you so much for watching this video till the end. Goodbye for now. See you in the next one. Before you go, I want you to do a few things. Number 1, subscribe this channel right now. The more you subscribe, the more valuable pharma insights and expert tips and industry updates we can bring to you. Also, please comment your feedback. This will really help us to improve the content, quality of the videos, and many things. And finally, share this video to at least one person who you think this would really help them. Thank you so much, and I will see you in the next time. Until then, goodbye for now.